Thank you very much for this kind introduction. It's sort of, I'm honored and humbled, and the only thing that the students in the audience really should know about me is that I love doing science, and this is really what you should remember about me. I'm just a scientist. All of the other stuff just happens, and I still don't know quite why it happens to me. So what I'm going to do tonight is something different from a normal scientific talk. And what I like, in order for you to understand why I'm doing this, has to do with the fact that the last 9 months I spent on sabbatical away from the lab, which gave me the luxury to think about how we understand scientific practice today and what I think is important in this scientific practice. So the reason why it's called the art of border crossing has to do with what I call integrative multidisciplinarity. And the reason why this is important is illustrated on this particular slide. And for those of you in the audience who come from the arts or are literary scholars, you all know that one particular subject can be represented in lots of different ways. And I'm trying to get some music up here going, and I was told it would work, but it doesn't. Okay, you can all hum Smetana's piece. Can you hear something? I can hear a tiny bit of the Moldau. And what I've put on this slide is a photograph of Krumau and a Moldau, and then there is a series of paintings by Egon Schiele, who represented this particular village in lots of different ways. So one particular object can be looked at from very different perspectives, be it a musical perspective, a photograph with pixels, or a piece of painting. And this is what I believe is also applicable to the natural sciences. If you consider physics, chemistry, and biology, no single methodology or perspective sees all of the phenomena of a particular object that you're looking at. And what you need to do is that you have to combine those different perspectives to try to get a more complete picture of what you're actually investigating. Just as Dr. Faustus says, what you want to understand is what the world is holding together in its entirety. And you cannot do it from individual perspectives. So there is quite a lot of anecdotal evidence that this is sort of true. Whereas in the 17th and 18th century, there was the lone researcher who had a brilliant idea, as depicted here in the Rosenquist pictures, where they come up with this wonderful thought and something new comes about. In the 21st century, science is done in a different manner. We all know that there is lots of anecdotal evidence that people from different fields together can combine their knowledge and jumps in science come about. Like Rosalind Franklin, a chemist, Maurice Wilkins, a physicist, Jim Watson, a biologist, Francis Crick, a chemist. All of those led to what became one of these very big new things in biology, the structure of DNA. And it took all of those different perspectives to come up with a picture of DNA. So although we all believe that interdisciplinary research can lead to big breakthroughs, we basically have only anecdotal evidence for that. However, there is this whole policy field, whether it's in Europe or in the US, that pushes and tries to foster interdisciplinary research. When you actually look in the literature to find systematic and hardcore evidence whether the effects of the research outcome of such interdisciplinary research really has a qualitative, much bigger impact than other research, there's very little known out there. There is also a lack of consensus how you would measure such effects. And for those of you who sit in the audience here, you know that collaborations are actually carried out by people, and they can be very fickle in their likes and dislikes. So yes, institutions can support interdisciplinary research, but in the end, it's up to the individual researchers whether you carry out such work or not. Now let me start out with a few definitions, because when we talk about disciplines, lots of people have different ideas what a discipline is. The word is actually derived from the Latin word disciplina, and it was used in the very earliest sense as an instruction giving, or meaning, the meaning was the instruction giving to a disciple or to a pupil. And you can really think of disciplines as these autonomous thought domains where you organize the thinking around a problem, around a theory, or around a method of investigation. Now, the reasons why disciplines emerge have to do something to some degree with the nature of human beings, because human beings have a natural tendency to separate, classify, and then conceptualize the world, because it makes it easier for people to come to grips with things. And also because there is all of this knowledge that is accumulating, and to take advantage of it, we try to compartmentalize it to some degree. Now, I found it interesting to look at how disciplines were characterized. And if you're in Europe, the term was used much, much earlier than in the US. And this already tells you that what we classify as certain disciplines has to do a lot with the cultural context of how the term is used. For instance, in Europe, it's been used since the Middle Ages, and it was really then used in the sense as we use it today when the first universities were established, Bologna in the 1100s and Paris in the 1200s. Interesting, when I looked through the literature, in the US, the discipline term only gets attributed to the period 1870 to 1900, much, much later. But coincidentally, this is also the time when most in the universities in the US were established. And the way in which the different disciplines are portrayed are very much different in different cultures. So here I'm just looking at the Western world. If you then look at the Eastern world, again, the way how we look at disciplines and categorize things in the sciences is quite different. Now, talking about interdisciplinarity, there is also quite a range of different terms that is used by different people. And all of those terms I would surmise under the term of cross-disciplinarity. There is what is called multidisciplinarity, and this is basically you look at a particular subject from different points of view, basically using different disciplinary perspectives. Then there is interdisciplinarity, which basically, in which the study ends up with a unified theoretical and conceptual identity. So during the study, it's forging a new identity rather than looking with different identities at a particular subject. And then there is also the term transdisciplinarity, which is very much pushed by Helga Novotny, and she likes the transdisciplinarity term because she always claims that it has the sort of the connotation of transgression, and she likes the idea that you have to transgress to make things new. So all of those are used, and what I really like best, and I think what represents to the best of my knowledge to what's going on in the sciences right now, I would call integrative multidisciplinarity, namely that we look with different perspectives at a particular subject, but then we integrate over all of those different perspectives to try to get a coherent picture. Now, the people who link different disciplines frequently come from hybrid disciplines. And what hybrid disciplines are, I've illustrated on this particular diagram. Just think of the traditional disciplines: chemistry, these are these purple squares; biology, the blue diamonds; physics, the green pentamers. And if you look at the boundaries between them, then you can see how those new disciplines emerge. Probably the one that's the oldest is biochemistry at the boundary of biology and chemistry. We have biophysics, and then we have a whole other group that all emerges from the boundaries: chemistry and biology, chemical biology, physical biology, and so on. What I want to illustrate here is that those boundaries are not fixed at all. That's why we have these broken circles. And they're constantly recreated and reshaped, because whenever something goes on at those edges, then new ways of looking and new reshuffling in those areas occurs. And the knowledge that is created by these hybrid disciplines, again, reshapes the whole landscape here. Now, those fields, as the hybrid disciplines that arose by the division and recombination, are different from what are traditionally the subspecialties in the different disciplines. And I've only done this here for chemistry, where we always have had analytical chemistry, organic chemistry, inorganic chemistry. And that's very different from those new disciplines or fields that emerge at the boundaries. And it is still really necessary to have traditional disciplines, because they really constitute the teaching domain of the sciences. And the reason why we need this, and to educate people in traditional disciplines, is that there is really nobody, or hardly anybody, who can apply the very sophisticated methodologies that are inherent in the different disciplines at the same time with the same degree of rigor in the same subject. So just because we have an enormous wealth of knowledge and we have an explosion of new methodologies, we have to make sure that whoever is trained as a chemist still will learn all of the necessary tools and background to apply the traditional disciplinary investigations in a very rigorous manner. Now let's look at a few of those new disciplines and fields that emerged. You can really follow the birth of those fields by looking in the literature. And for that, you can look at the most of the scientific work is basically disseminated by publications in the literature. So you can follow those articles in the literature and you can really see when new fields emerge. For instance, structural biology as a field basically only appeared in the 70s and 80s as an independent field. And I myself and lots of people who are now called structural biologists, we were never exposed to biology when we studied. We did chemistry and physics and then ended up applying our methodology to biological questions. Bioengineering is another of these new disciplines that came about in the last 2 decades only. And what I've shown on in this diagram here is showing the birth of what's called neuroscience that emerged around 2005 as a standalone discipline. And you can actually see, just following citation patterns in the literature, that there were 2 independent branches, neurology and neurobiology. And by now, it's all one discipline that's called neurosciences. Now you can go and look at those flows of citations and how the different fields and subdisciplines hang together in diagrams such as those. And this is a diagram that represents different disciplines at one particular point in time, 1974. And the different fields are color-coded. Chemistry is blue, biology is green, biochemistry is cyan, and bioengineering is magenta. And then you can look at the net flow of knowledge, as it's called, but it's really citations, from one area to the other. And at that time, biochemistry had really the largest flow in and out of different fields of the natural sciences. Bioengineering had almost no presence in biochemistry at all. And if you now fast forward 10 and 30 years to 1984 and 2004, you will notice that the role of bioengineering not only had increased in biochemistry, but it's also now in different other fields like microbiology. And so what you can do in that sense, see how interconnected certain fields are and how the influence of one of those areas, chemistry, radiates out into different scientific fields. However, if you do that, you only know that chemistry feeds into biochemistry and vice versa. But you don't really know what types of research is going on at the interfaces, because this is really done at the citation level and the journal level, not the individual article level. And what I was looking for is getting some idea is how one would follow the different merging and recreation of new fields at a more fine level, at the individual article level or at a group level. So what I thought is that those types of breakthroughs follow nonlinear patterns, and what one really should do is look how those different disciplines emerge and come together via different teams. Again, what you need to do for that is look at bibliometric data, and that can be quite difficult. And again, doing this, you need sort of network models, and probably what you also need is a combination of statistical and stochastic simulations to try to find out what's going on. And I apologize that this is cut off here. Probably we should have checked out the resolution of the screen before I started, and we didn't do that. So what do I mean by team science? Team science is really when two or more research groups come together to solve what we call a complex or multifaceted problem. And the reason why those different groups come together has to do with the fact that we have a vastly increased range of questions that we are asking now in the science, and that the difficulty of the questions is also increased. We have large data sets that we really have to analyze, and we have very big advances in technologies, so that if you are a hardcore NMR spectroscopist, you may be very good at what you're doing, but if you need another methodology like cryo-EM, yes, I can dabble in it, but I think what is much more powerful if I team up with somebody who's really an expert in cryo-EM, and then together we can solve the problems more effectively. And if you just look at the literature and look for the term interdisciplinarity in articles, you can see it's sort of really, and that's not just in the sciences, it's really a term that's become exceedingly fashionable over the last few years. Now, what it really also reflects is that there is an enormous increase in the amount of science output that is generated. So we can, if you look at interdisciplinary teams, we can hopefully speed up the rate of discovery. We can apply novel methods, because it's only when we have novel methods that then get put to use at a problem that the old methods don't work, can we move forwards. We have this enormous increase of knowledge and facts that we have to deal with, and then we have to apply specialized knowledge to look at maybe emerging problems. And this is a particular database, and you probably know it, Scopus, that at the time here, I think it's, let me look at my computer, 2010, that's cut off here. There are 30,000 journals that just reflect those 15 top disciplines in the sciences here. Largest chunk is the medical sciences, but then physics, engineering, molecular biology, chemistry. So this is an enormous data avalanche, and they all report on scientific knowledge. Now, why is it, or why can one understand that maybe working in teams is better than just working in what I would describe here in a linear type of arrangement? If you have a team that is that combines all of the same types of people, so put 4 NMR spectroscopists together, you have what I would call a shared mindset. And what happens is that, yes, you will add all the knowledge up, but you will only get incremental advances. And this is reflecting the sequential type of thinking. If you have networks in a team where you bring together people with very different methodologies and perspectives, then you have this connective thinking going on. And in the center here can be a member of the blue team at some stage, or a member of the green team the next week, or a member of the purple team, or of the golden team. So this is really a very fluctuating and dynamic system. And in this way, you have a much more connective type of thinking going on, which can lead to more radical advances in science, I would argue. Now, if you want to look at this in detail, you have to look at all of those databases. And there are lots of those databases of the scientific literature around. There is the NSF collection of bibliometric data, and they currently, and currently means 2010, cover roughly 5,100 journals. There is Thomson Reuters, which you all know as the former Web of Science, and they have 12,000 journals. JSTOR has a lot of journals, but there are very few science journals. They have biological sciences in there, but no chemistry and physics. And then there is Scopus, which is a big database that's combining all of the publishers together, and they have lots and lots of different articles. So if you look at the NSF database and compare 1988 to 2010, you will see that the science that carried out in team is becoming more and more prevalent. If you look at the overall output of co-authored, and co-authored means they are not two different people, they're two different institutions, articles, they have grown tremendously, and I think I've got the growth here. In the world, those articles grew from 40% to 67%. And internationally co-authored, and again co-authored means different institutions, grew from 8% to 24%. So basically you see a steady increase in what I would call team science, with groups of people from different institutions coming together. And you can see this is true for all over the world, not just for the United States. And also if you look at developing or countries that are less advanced in the sciences, for instance China, you can see that their co-authored articles really go up. So they now combine a lot of Chinese science with science somewhere else. Let me show you what I think is a real success story when it comes to team and interdisciplinary sciences. This is a program called the Human Frontier Science Program, and it's a program that selects people who put forward teams from different areas of the sciences. And they're selected not only because the individual team leaders are of very high scientific standing, but also because what they wanted to do is really foster this interdisciplinarity. The program was founded in 1989, and up to now it's funded over 900 collaborative teams. They also fund postdocs and junior investigators, but I only looked at those collaborative teams. I had access to all of their records, applicants and awardees, and I have all the publications that are linked to individual articles. Now, the program first had a strong emphasis in the neurosciences, and that's why you can already see that it's been quite successful. The Kavli Awards, these are sort of the equivalent of a Nobel Prize for the neurosciences. 7 of the awardees got, of the up to now 9 of those awards were Human Frontier Science one. And 18 of the grant awardees went on to win Nobel Prizes. So this really has been very successful. Now, if you look at the interdisciplinarity of this particular program, the disciplines that are involved are life sciences, like sort of the more the medical sciences, chemistry, physics, mathematics, computer science, engineering, and others. And when it started out, there were basically very few that had strong interdisciplinary teams put together. However, this has been steadily going up, and now it's even higher than it was. 2009 was the last data I looked at. So basically, almost 80–90% of the teams have people from physics, chemistry, biology, or medicine in one of those applications. So this really shows that working in these interdisciplinary teams can yield high impact research outcomes. The other way you could sort of argue, just look at what Nature and Science does at the end of the year as their breakthrough of the year or the noteworthy research of the year. Those get published by Science every year, and I just picked the one from 2012, which was after the Higgs boson, where Higgs got the Nobel Prize this year, the runner-up. And this is a collaborative team that has 88 authors from 22 institutions. And this had to do with the first use of the free electron X-ray laser, which may probably revolutionize crystallography. And it was interesting when I first learned about this that it was thought that if you would use these very high laser pulses, that you would destroy your material before you could collect data. And it was actually maybe some stupid experimentalist who said, why don't we just try and look? And when they looked, the data was still there. So all the predictions would have said, you can't do it because the material is destroyed before you collect the data. However, it turns out that it wasn't. And again, this is something that will only come about if you have somebody from a different area ask a question that would never occur to somebody who has already predicted it's impossible to do something like that. So again, different perspectives help to solve problems. Now you can go even further. You don't have to just go to your scientist across the border in the next discipline. You can go out to the public and do the same sort, or use the same sort of approach. You can go out and ask whoever wants to contribute to contribute. This is an example where Tim Gower set up the Polymath project. The project at the University of Cambridge, and it was a blog where he put out very difficult mathematical questions, and they were trying to see how they could solve them. So they just give it out there. Within hours, mathematicians and teachers and other people from all over the world chimed in. And in just 6 weeks, they solved their first problem that way. Basically, by letting the public or any other researchers who wanted to contribute to solving that problem come in. Likewise, a problem out of the life sciences is being solved by using computer games, and anybody can play that game. It has to do with designing RNA molecules that have a particular function. And you can do all of the predictions on the computer by people who calculate the thermodynamic stability of the RNA and all of that and let the computer do it. What these people did and still do, ETE RNA or Eterna, whichever way you like better, they let people play a game and say, look, try to design an RNA with a particular function. And lots of people, school children, high schoolers, students chimed in. And what they promised the people that they would actually make the molecules and test them. And what was found out that through this participation of the public, they learned faster how to make useful molecules than if they would have just let the computer design more molecules. So I think this again shows you that opening boundaries to your outside world as a scientist can contribute to you getting faster and maybe better answers. Now, why was I interested in anything like that? I think it has to do with the fact that I'm called a structural biologist, and I told you I really didn't know much about biology. I had to learn biology while I was trying to address questions in biology. And what structural biologists really want to do is try to get a comprehensive picture of a cell or of an organism spatially and temporally. So we want to know where things are and how they move at in which time frame. And we also want to then use this to get some explanation for biological function, be it activity, recognition, selectivity, and we also want to do this at the atomic level. So if you look at the whole range of biological objects that are in front of us, they span a huge scale. Just thinking about the linear scale going from, let's say, an amino acid from 1 nanometer to 1 billion nanometers. This is a ginormous scale. So immediately we will realize that you cannot use a single methodology to look at all of those different objects. You all know that if you look at an organism like a human, you can open your eyes and use light and you can discern whether there is one person sitting in the chair there and another there. However, if you go to much, much smaller scales, you have to get some help. And you can get help from a microscope, which still uses light, and which stops at the, let's say, 200 nanometer level. So you can basically look at a single cell, a bacterium, or an immune cell, a T cell, under a microscope. If you want to look at things that are smaller, light can no longer resolve those objects, and you have to use different methodologies. And there are basically 3 structural biology methodologies around that allows you to look at molecules or objects at this scale: electron microscopy, NMR, and X-ray crystallography. And I'm not going to teach you anything about those methodologies. Take my word for it that we can look at those objects. And we have different requirements for the subjects that we look at. If you do crystallography, you have to get a crystal. And you have to then have a good single crystal that diffracts. NMR is done in solution, so you have to have a protein or a molecule that is soluble in solution in aqueous buffers, for instance. Cryo-EM looks at frozen specimens, and if you want to look at aggregates or amyloids, you again have to use a different methodology, namely solid-state NMR. And again, ideally, if you want to describe a system from all types of angles to the best of what's possible, you better use a combination of all of those methodologies. So why is it important that we get structures? You could argue, who cares what a structure of a molecule looks like? I just want to know why I have diabetes. Let me tell you that structures are very important. We can use all of those structures to get some important, detailed information about what's now a very fashionable field, namely systems biology. In systems biology, people make all of these very intricate networks. But what you really want to know is that this blue part of a protein sits next to the red part, because then you can really say they talk to each other and they influence each other. Structures are also very important if you want to understand a biochemical mechanism. For instance, in an enzyme, if you want to know what's the chemistry that goes on in an active site, you better know where every atom is that does the chemistry, and from that you can infer the mechanism. Drug design is a very important field, and what you need to do if you want to design drugs in a sort of semi-rational way is you can use structural knowledge and base your drug design on those structures. And I've taken here ritonavir, which was one of the first very effective HIV drugs, which is a protease inhibitor. And this particular inhibitor was basically designed totally on the knowledge of the structure of the HIV protease. And then we can also learn something maybe about diseases. The example here is the protein called p53, and in a lot of cancers, and in particular in a lot of prostate cancers, they have been mapped to mutations in the p53 molecule. Now, one domain of the p53 molecule is a protein that recognizes DNA and is a transcription factor. And if you look at the hotspots of the mutations in those cancers, they basically map to this domain, and they really map to those residues that interact with the DNA. So clearly, if you have a mutation of those recognition residues in that transcription factor, it is easily understandable why this particular protein can't do its job anymore. So that's why we need structures. And then we have to take the structures and combine all of our structural knowledge with biochemistry, cell biology, spectroscopy, and also in vivo studies, because we want to really relate what we see in the test tube to what goes on in the organism. And sometimes we can't get all of the information we want from our experimental data, and then we can resort to computation, and since we now have a computational Nobel Prize, it really puts the computational biology where it belongs, namely there with all of those experimental techniques, because the combination of all of those methodologies will allow us to try to answer questions not only on the overall big scale, but what we really want to know, for instance, if this particular hydrogen bond is very important for a particular function, and for that we need our atomic structures. Now, I'll give you one very brief vignette of how I try to implement my thoughts about working together in multidisciplinary teams in the next, I think, 2 or 3 slides. What we have in Pittsburgh is a center for HIV protein interactions. And these centers were established by the NIH to try to understand some more of the basic biology of HIV, because although it's been 30 years that we know that the virus causes AIDS, we still don't have a cure and we still don't have a vaccine. So basically what we need to do is carry out some more basic research to understand how the virus can wreak such havoc with a human or a mammalian system. So what we are focusing on in our center are what called the immediate post-entry events. These are the events after the viral membrane fuses with the cellular membrane and the contents of the virus gets released into the cytoplasm. And it's sort of this empty blue lake here, but you all know, if you know a little bit about biology, that the cytoplasm is a very crowded space. There are lots of molecules in there. And clearly what happens there has a lot to do with the normal cellular biology. And the virus has to overcome what normally would be the innate immunity of an organism, trying to get rid of those invaders. So in the center, we actually have medics, biologists, virologists, microbiologists, chemists, biophysicists, engineers, and we all try to work together to address questions how the virus actually negotiates this rather unfriendly environment till it ends up in the nucleus and can integrate its genome into the human genome. I'll give you one example how we all work together to get an answer to a particular problem. Let me go back. This particular shell of the virus is called the virus core or capsid. It's made of out one protein, and what we wanted to understand is how this protein makes that shell and how it protects the virus from going through here. The protein is a relatively small protein. It's got only 340 amino acids. And what we did by NMR is determine the solution structure of what is sort of the building block of the protein. It makes a dimer with its C-terminal domain. And this is this C-terminal domain dimer. You can see here it's by all criteria a really good NMR structure. You see that we have a nice hydrophobic interface here. What was interesting to see is that this interface at the end has one particular residue, tyrosine 145, which also makes an intermolecular contract from one monomer to the other monomer. This was sort of surprising. However, for those of you who are NMR aficionados, what we can do is map out very, very precisely how this particular residue sits amongst all of those other hydrophobes in the other unit by NOEs. Now, if you're a structural biologist and you see something like that, you immediately would say, oh, this has to be important, right? Why would it be there? But you can only say that if you disrupt it and you can show that it's important for the biological function. Otherwise, it's just a structural biology curiosity. So what we did, we took this tyrosine and changed it to a alanine and to a phenylalanine. If you do that, the virus is dead. So now we really know that this particular amino acid is very important. And for those of you who are not chemists and you don't know the difference between a phenylalanine and a tyrosine, it's a single atom. It's missing that oxygen. So one atom difference in 2,500 atoms makes the virus dead. So small differences can be really, really important. And this is really shows you also the combination of doing structural work immediately interfaced with the in vivo work of the virologist, because otherwise it would have just been a structural curiosity. Now, we've pushed this system further, and for those of you who want to listen to me tomorrow, if you can't get enough of me tonight, we now have really looked at what a capsid looks like in the cell. For that, we first started out with the individual structures that were done by NMR for the capsid protein. Then there was in vitro assembled capsid that makes long tubes, and you can do cryo-EM on those. And then there was in the cell cryotomography done, and cryo-EM tomography is basically like a CAT scan on one of those capsid cores rather than on a human. So what we then did, assembled this all-atom tubular model from which we knew what it looks like in terms of the hexamers. And then we fitted this to the outline of the tomogram. And although it was well known that you'll have a hexameric lattice, and that if you want to close a hexameric lattice, you have to put pentamers in, it wasn't clear where the pentamers are, and up to now people have made only very symmetric mathematical cones. So what was done here, looked at the outline, and we looked at where are acute curvature changes. And in those positions, we put pentamers. And then with that, we ended up with an all-atom model of the capsid. And I'll just show you a little movie so that you can look around there. So this really is a combination of NMR, crystallography, cryo-EM. And to put all of this together in an all-atom model, we needed a lot of computational work as well. So here again, it's the combination of all of those methodologies that gave us the final picture. And I'll stop with that movie and just go on. So in the last few minutes, I want to try to explain to you why it's not just my pragmatic way of looking at the integration of the sciences, but rather a very basic philosophical understanding on how we look at the world that tells us that we need this integrative multidisciplinarity in the sciences. We all believe that all matter that's out there is made of the same type of stuff. We're all made out of atoms, right? However, if you look at complex systems and we try to use different representations, by the sheer nature of that, those representations have to be partial. They can't be completely encompassing the real entity. So we need these multiple representations in order to get at the picture of what the world looks like. Some of the reasons why this is the case are listed here. Complex structures can arise from multiple different sources, and different components can contribute differently to those. The behavior can also be influenced by different factors, and sort of an overall behavior can be the result of 2 or 3 very different underlying features that come together. And this is what people call robustness of systems. And then the environment plays certainly a very important role, and the context in which a particular object would reside will influence how the object behaves. So again, if we just look at one perspective, we lose out because we simplify the view and have to, by necessity, leave certain things out. And sometimes the way you do your experiment also prevents you from seeing the entire system. Like if you do an NMR experiment, you work on an isolated protein, but we all know that the protein in the cell is not an isolated protein. So just by the sheer methodology, we know we are incomplete. And therefore, we really have to combine all of those methodologies. And I want to sort of finish up by using an example that comes from Kendrow's Nobel Prize lecture. And we probably all believe in Anfinsen, and this was very shortly after Anfinsen found out that if you take ribonuclease A and you denature it and you put it in the test tube, it can renature and fold into its normal shape. And what Kendrow said, that if this is really true, and if we understand proteins and we believe that the structure of a protein is really governed by the intrinsic property of the polypeptide chain, then at some stage X-ray crystallographers will be out of business because we can predict what a structure looks like. So why aren't crystallographers out of business yet, 50 years after Kendrew said this? Actually, they're doing extremely well. If you look at the PDB, it's predicted that January of next year, the 100,000 structure will be there. Even NMR, we've just reached the 10,000th NMR structure. So experimental structures are done all the time. And we haven't lost out to the computational predictors yet. Lots of methodologies now also contribute to getting structures. So the reason is exactly what I alluded to before, because when we look at a protein by crystallography or by NMR, we only have a selected view of what the protein looks like. And if we would completely understand how an atom ends up in this position by just looking at the final structure, yes, we would be out of business. So if you think about it, we have proteins that live in these different habitats in the computer where people do these predictions, or in vitro where NMR or crystallographers determine structure. But then in real life we have them also in the cell, and there again they behave and look differently. And it's really only when we combine all of those different perspectives of a protein and the structure that maybe in the end we will understand proteins. I thought 10 years ago that by now I would understand proteins, and I still don't understand proteins. And it's really back to the original picture. We can look at a photograph, we can look at a painting or the score, and they all reflect this little village at the Moldau. But they're very, very different in their views of the same subject, and the same way we look at things with different views, whether it's computation, crystallography, or NMR. And we really need to have all of those perspectives and then get together and integrate them to understand what the world looks like. And I think I should really finish off because I'm over my time. Let me just give you sort of the last philosophical bit. We look at one object, let's say proteins, with different methods. We end up with different models and representations. And because all of those only give us a partial view of what a protein really is made of and what it's like, we really have to integrate all of that. And it's not a just additive viewing together. It's always a two-way street between the different perspectives, and one informs the other. The computational people learn a lot of what the proteins look like, and we learn from the computational people what is a sensible way of representing structures. So all of this has to come together. So what I really want to tell you here is, at the end, if that is really the case, if we as scientists work in teams and we believe that this is the correct and the most complete way of addressing and understanding nature, why on earth don't we let PhD students work in teams and try to get them also to learn from each other and integrate their knowledge? I have to say, I tried very hard in my environment, but departmental boundaries don't seem to be as flexible and transparent as I would like them to be. But maybe the students and the next generation can push for that, because if we don't have the students, we don't have any future for the science at all. And if I have convinced the students that this may be the way of how science should be done in the future, maybe they will take it and run with it. With this, I really should tell you all of the people in the team that contributed at least to what is going on in the Pittsburgh Center for HIV protein interactions. Jin Wu makes all the proteins, and for the crystallographers, every protein is N15 labeled because we do our quality screening by NMR. We can see whether it's a good protein, and then it gets put into drops and set up. Inja runs the NMR and checks it all out. Pei Jun is the cryo-EM person. Chris is the virologist who does all of the in vivo testing of what we sometimes think is important. And Klaus Scholten did all of the huge computational work for the oval capsid. And I didn't talk about solid-state NMR or some other crystallography, but those two people are also part of the center and work together with trying to get us answers that are better than each of us individually could do. And we are very lucky that we have funding not only from the University of Pittsburgh, who put all of the instrumentation there, also General Medical Sciences and the AIDS reagent program gives us money to look at the AIDS, the HIV work. However, all of my thoughts about integrative multidisciplinarity, I was very lucky that the last 9 months I spent at the Institute for Advanced Studies in Berlin, where I just could have the luxury of thinking about how I like science to be done and maybe how it should be done. And a colleague of mine in the Department of Philosophy of Science in Pittsburgh came to visit me there, and we chartered some of the thoughts that I presented today. Thank you. I have to look back. It didn't look to me that it flattened out. It's actually probably still going up. The first question, that's really what I wanted to do in Berlin. And it is enormously difficult to really do that because you have to do the linking at the individual article level, which becomes very difficult to do. Just thinking about that there are 30,000 journals already in the database, and just think about how many articles there are. And that's why in the end I went to the database of the Human Frontier Science program, because there I had all of the articles linked with individual researchers and teams, and there I could do that. I still haven't completely given up on looking at the question in the big picture. I haven't found anybody who's done it. I was hoping that maybe somebody had already done something of that type. No, it hasn't been done. So I'm hoping that maybe if I team up with some of the publishing houses who have a vital interest in finding out whether their papers are high-impact papers, but impact in the good sense, maybe not in the sense as in the last issue of Nature on the front page if you haven't read it yet. So I think me as an individual person, I was not able to do that because the data amount is so ginormous that it was impossible to do. And that's why I went in the end to the Human Frontiers one.
So you talked at the end briefly about graduate students needing to get more involved in this interdisciplinary type of research. There must be some level of training that would go on as an undergraduate to prepare it for being exposed to all these different methodologies, to all the different parts of the system. So, do you think that the major university systems now are doing a good job of training undergraduates for this type of research in the future? And if so, that's great. And if not, what changes do you think need to happen?
I do believe that we need really good training in the traditional sciences, because you don't want to become a jack at all trades. What you really want is to be very good at some particular methodology or very good in one particular discipline. But while this is happening, it would be, in my view, a good thing if you would, in sort of particular questions, team up people like a chemist, a physicist, and a biologist, and let them work together on a problem, because they will look at the problem differently. And I think whether it's done in the last years of undergraduate, when they do, some people do a particular project or a senior honor thesis or something like that, or whether it's in the early days of your graduate career, I'm not sure whether it's important exactly where the timing of it is. But overall, if we as researchers collaborate and work in teams, I think it would be good for students to learn to work in teams. And what I've seen, I've heard the argument that from some of my colleagues, they say, oh, but how do you then know who has done what? And I've teamed up people, and I've seen if there's 3 people working together, there's a dud in the team, this person is gone very quickly. The students don't tolerate sort of people who go for an easy ride. Yes, they do, and you can do it, but it depends what time resolution you want. If you don't want really fast times, certainly I don't think NMR would do it. Maybe femtosecond crystallography can do it. You have to get a lot of different snapshots. But I think in the real fast time regime, it's really spectroscopy that allows you to do this. But spectroscopy doesn't give you structural resolution at the atomic level. So integrating, again, those methods with the structures is what's needed.
What do you think about, you talk about scientists working together, how about humanities being part of teams or expanding ideas and networking even more to bring in different perspectives?
I think depending on the questions, and if you think about big questions that are important for humanity, like what are we doing with the energy crisis, or let's say transportation in this country, I think there are people from other disciplines, not just the natural sciences, that can be playing a very important role. Yes, indeed. if it's sort of biochemistry and bioengineering, I think you can do it. If you want to be an astrophysicist and a geneticist, I think you'll have a slight problem. So, but I think it's a really good idea to sit together with people that come with a different mindset. It's sort of like you have in the graduate program, you have the physicists who come in and you say RNAi, and they look at you and they have no idea what it means. But if you then talk about some wavelength behavior to the biologists, they have no idea. But when they sit together and have to solve a problem, they actually are much better teaching each other the language than maybe us teaching them. Okay, the Salk Institute comes to my very first slide. People are fickle. Institutions can tell you whatever you want to do. If you don't have two people who really want to work together, it doesn't work. Protein deposition diseases. There, I think it's a very interesting way to look at this with a philosophical framework of pragmatism and understanding the molecular basis of it. We know that, for instance, if you overexpress like chaperones in the cell, we can, to some degree, alleviate some of the aggregation of proteins that give cause to those protein deposition diseases. We still don't know exactly how it works. But having structures and trying to see how those initial structures could be prevented by having chaperones around will probably help us. My personal view is that what we see as the tangles and depositions is probably the end product and not necessarily the toxic species.
Because this talk about the interdisciplinary cooperation. What do you think about like cooperation with different institutions that are working in the same field? Like, I think most of the people who are doing PhDs here are aware that someone else on the planet is probably doing the same research for like the same amount of time. After 4 years, before you trying to publish, somebody else has published it. It's a waste of money, time, stress. Do you believe if there is a chance that somewhere in the future, like institutions, different countries, to like build up a network, to like all the ideas to be put together and establishing cooperation between like the same field, but between different people?
So this would be like, we all want world government, right? And it's sort of utopia. I think right now, the thrill of going after something that is really important and we don't know entices people to really give their best to get an answer. And you don't know whether person X over there is doing the same as you. And the only thing that, it's sort of tongue-in-cheek here, you don't waste your time. You can work on something that's so boring that nobody else will be working on it, and then you have no competition. If you work on something that's really, really interesting, there will be competition.